let's expand on your presence in the Epoch Times documentary, which was an hour, basically describing the backstory of how this virus was engineered. You referenced that what we know now that it has a protein from HIV. I think it's the envelope protein, the GP41, that's in there. And then it's also got SARS, which seems to be engineered. And I mean, the studies that they cited were, I mean, all the way back from Dr. Shi in 2010, 5 years before the 2015 North Carolina study. So she's been working on this a long time. So she's at the Wuhan virology lab. So tell us the story from your perspective of how this was put together, and then integrate that into the fact that this was put into the previous flu vaccines. I mean, you're putting all the pieces together now.
Right. And it's really just, it's not put together as in cut and paste, which we call, we call it
CRISPR. CRISPR.
Yeah, well, there's CRISPR, but there's also simply pseudotyping. That's when you express the envelope of, let's just say, HIV, because that was what was found, both GP120, and then later that paper was forced. The investigators from India were forced to retract that paper that was in that documentary on March 5th. But I knew that our colleague Luc Montagnier, the Nobel laureate, had similar data and also had found GP41, which is the transmembrane domain. So it's important because people say, well, she said GP141 and the paper said 120. Well, and then it also had the folks from India also had GAG. That's kind of structural proteins. So that gives you a little bit of clue that it wasn't a CRISPR technique or a pseudotyping where the envelope was expressed in a gene, in gene therapy type of way. But what was done is the envelope, the virus was acquired as they grew SARS-CoV-2 in Vero E6 cells. So the monkey kidney cells where you get HIV. Simian immune deficiency virus was the origin, and we were told all the way back in the 80s that somebody forgot to cook their food in Africa and a few promiscuous men spread this virus around the world. So you can see again the patterns of the lies and of what people believe. So I believe it was simply in the data support, because if it were CRISPR, you wouldn't put the GAG sequences in there.
All right. So the technique is not really that crucial, but I'm curious if you could explain why they would want to do that. What purpose does putting an envelope protein from HIV into this virus do? Does it increase the infectivity rate? No, it doesn't increase. It impairs the immune system, I would suspect.
Absolutely. The first thing is with SARS-CoV-2, you must grow a virus to make a lot of it. So you grow it in cell lines. They didn't take it from the bat and it jumped into a human. It normally goes through another cell, a monkey, a smaller animal, a monkey, or among us, another species. They call them civet, C-I-V-E-T, and I don't know what that, I'm not a zoologist. So a small animal. So the cell line that supports the growth and expansion is a monkey kidney cells. So for what purpose? Maybe it's not engineered at all, and I wouldn't use the term put there. But yes, the end result is now you don't only infect the epithelial cells of the lungs, you infect the white blood cells. You infect the immune cells. We see the splenomegaly in large spleens. We're seeing penias, cytopenias. We're losing cells like HIV killed T cells, and that's why we started there. So it's got not only an expanded host range, but disease symptoms that make no sense for a coronavirus. Hence, we're killing people because they're treating an upper respiratory infection, and you're getting that inflammatory disease signature because you're infecting the very innate immune response, the macrophages, the monocytes, the natural killer cells, the T cells. That's it, and it's primarily the T cells and the macrophages, because that's where HIV 120 and GP 41, those are the cells they infect through CCR5 and the CD4 receptor. So now you're going to lose your adaptive immune response. You're going to drive the inflammation, the fire higher, and it's the fire that does the tissue damage. So to what purpose? Yes, it makes it far more pathogenic.
So there's a lot of other elements to the pathogenicity, and one of them is the construction of the spike proteins which bind to the ACE2 receptors.
Correct.
So it appears that from the papers that in the Epoch Times documentary that was engineered. So Correct. Can you comment on that? And I think you even gave a little comment in the documentary.
I did comment on it. Yes, clearly this came from SARS. The original SARS, which also infects through the ACE receptors, and there are some single point mutations there that make it far more infectious, easier to spread, and how those were acquired, nobody really, can say, because in the studies that have been done so far, and this is where people get caught in the weeds, because we've got an infection that has gone through many different hosts, many different environments, Switzerland, Sweden, 190 countries, where the immune responses are clearly different. There are different susceptibilities and things like that in different people, which is what we've learned over the decades. So those things change a virus. Every time the virus divides and infects a new person, it's by definition changed. I call it the Heisenberg uncertainty principle of biology. When you measure it, you change it. When you grow it, you change it by definition. So I don't like to say engineered. And at this point, so few sequences so few viruses have been fully sequenced, and even less so on March 5th, to where you can start saying, well, it's not that, and it's not that, and you're crazy, because there's no evidence of that.
Let's go to the other modification that's been commented on in conjunction with the chairman of the Harvard's Department of Chemistry, Charles Lieber, who was arrested late last year. He's an expert in nanosciences and was given a multi-million-dollar grant from China and NIH grants, or was being paid, cross-paid, which is why the federal government arrested him. Yeah. And he didn't disclose it, and Harvard claims they didn't know what was going on, which probably not true. But nevertheless, his expert, he was there for 8 or 9 years, right. And literally was getting like $50,000 a month as a salary. So right. His expertise was nanoscience. So how does that fit into the picture? Was it done? How did that integrate into the process? Was it able to aerosolize it, or for transmission, or what's your thoughts on that?
Correct. Both aerosolized it for transmission, a lot more transmits than a normal coronavirus. So they are aerosolized, but the nano further increases the host range. So now you can go in every cell. So now you can go across the blood-brain barrier. That's nano. Now you don't need a receptor. So you've aerosolized it, you can breathe it, it can go into your every cell in the body. You don't need the gatekeeper, you don't need the receptor, you don't need the lock and key.
Yeah, that makes perfect sense. So now can you tie this all together and into the amazing fact you revealed earlier, where this was actually in the earlier flu vaccines? So how did that connect? Because I've never heard that connection before.
Because they're grown in animal cells, and because they have some of the same viral proteins and lock and keys, and as they're floating through the laboratory, where you're growing up in large stocks of these cells, aerosolizing them, it contaminates and cross-contaminates through the air. And this is what we found, and so what do most people get? Oh, they get a flu vaccine. So if those vaccines, and this is what we found in 2011, the big, oh my God, was we can't afford to retrofit our laboratories and our manufacturing facilities towards biosafety level 3 and 4 to protect the lab workers who are spreading these viruses, getting infected, and now they're aerosolized. We've got contagious cancer viruses.
So
and all the cell lines are contaminated.
How do you speculate that the coronavirus got into the flu vaccine manufacturing facility?
If they're using the same cell lines that are used to grow.
Those cell lines were contaminated.
Yeah, the cell lines can contaminate it in our paper, in papers work that was done, which was what finally they shut us down because it
how did those cell lines get contaminated? Was it
by the air in only 2 weeks? If you take an incubator and you put one cell and you put the other, they cross-contaminate through the air in only 2 weeks. So just the act of housing them in the same incubator.
Okay, so it sounds like the problem started when they're creating the cell lines, where they're not making the vaccines. They're making the cell lines to grow the vaccines that they sell to another company to make the vaccines.
Correct.
So do you speculate this was intentional or was this accidental?
I don't think it's intentional. I never did, because we're not God. We're not. But simply, and this is what our book Plague of Corruption, really the message of it is. So the message of Plague of Corruption is we can't mix animal and human tissues, not just coronaviruses, but the infectious retroviruses. We are injecting lots and lots and lots of animal tissue, fetal tissue into humans, and we're creating novel viruses all the time, even within the individual or family.